want to say credit maybe, or not enough due during this class. Unfortunately, microbiology covers such a wide breadth that it's hard to fit everything in. So before we get started, Elena, could you just humor me and just kind of try to think off the top of your head, why is it important to know both the similarities and the differences between prokaryotic cells and eukaryotic cells? Why do we need to know that? Why does this matter?
I would say because prokaryotes, aren't they the ones with the nucleus? Therefore, they might have different functions, and you would like research them differently than eukaryotes.
Okay, so I like most of that. Which one did you say had the nucleus?
Wasn't it prokaryotes, or is that wrong? Eukaryotes.
There we go. Nailed it. Right. So eukaryotic cells, right, have a nucleus, and so one of the things that we pointed out very briefly between prokaryotic cells and eukaryotic cells that Elena has mentioned is this idea of membrane-bound organelles. And I really like what she said there, right? If you are doing research on them, it's important that you know similarities and differences, because that's going to adjust how you interpret the data or how you correlate things. All right. I said, prokaryotic cells are an excellent organism for us to use for research because they have a lot of similar metabolic pathways, right? We can grow them really quickly and really cheaply, but they do some things differently. And so you have to be careful what conclusions you draw about eukaryotic cells having a similar function possibly. All right, what else? How many of you are going to be nurses or doctors or PAs or PTs, right? Even to an extent. Why is knowing the differences between these or the similarities between these important? Like, don't overthink it, right? I keep my questions simple. Why is this important?
Well, is it like to differentiate, like, because prokaryotic is like bacteria and like the archaea or something, and then eukaryotes are like the eukarya, like, so it's like important to like differentiate between like what you're looking at, or I guess maybe that's the same answer as before.
It's kind of the same answer, but I totally agree, right? As we discover, especially as we discover new organisms that we haven't seen before, knowing those similarities and differences is important for us categorizing them. So I definitely think from a pure knowledge standpoint, I'm not disagreeing at all. I think that is really important. So no, I think that totally works. It does kind of fall in line a little bit with Elena's answer, but it's a little different too. So I like that. What else? Those of you that are going to be doctors or nurses or PAs.
Like when we think of like the human body, I mean, like if you think of cancer in general, cancer is like the growing of cells like uncontrollably where there's no like checkpoints or anything. And so then knowing like how it's supposed to work and then how if it's like messed up, like how you're able to fix that based on. So like looking at the two differences of how like a prokaryotic cell could affect a eukaryotic cell.
Okay. So knowing how they would interact with one another, knowing how they influence one another. Okay, I like that. That is definitely a kind of, in a way, specific big picture, right? So when you say that, I think of like the microbiome that we mentioned yesterday, right? If you guys remember from that video, the immune system was interacting with the good bacteria in the gut and helping to train it. And we'll talk about that a little bit more. So definitely understanding similarities and differences and how that influences the interaction between the two cells is really important. There's kind of one big thing that I'm looking for. One big thing left. You guys have said some really good ones, and I think these have been great answers. What's another big one?
Also, could it be sorry, go ahead.
I was going to say it helps us to understand how drugs will affect the different types of cells.
Right. So that is a really important one to keep in mind. All right. So we'll talk about this more, right? But when you take a drug, you want to kill the pathogen, not yourself. All right. And so understanding similarities and differences helps us do a couple of things. It helps us determine what's a good thing to target, right? So if at all possible, target unique features, right, of a prokaryotic cell that maybe a eukaryotic cell doesn't have. Also, right, kind of predicting, hey, I know this is going to affect a prokaryotic cell, but there's some things in eukaryotic cells that are similar, and so this is going to have a little bit of probably a toxic side effect. And so just being mindful of medications, being mindful of what we target is really important for understanding these similarities and these differences, right? And then, like you guys said before, how we classify them. If we're using them in a study and we want to draw correlations between eukaryotics and prokaryotic cells, well, knowing what's the same and what's different. And then, like Noelle said too, how do we classify them? How do we group these things together? Thanks, love. All right. So let's keep going. So what we're going to kind of do here is we're going to start with prokaryotic cells, and we're going to go from the outside and work our way in. All right. And this is really similar to an image that you guys labeled last night, or maybe even early this morning, for a prokaryotic cell structure. And where we're going to start is this area called the cell envelope. All right. So for the cell envelope, it has 3 components, right? One component that I hope you're all very familiar with is the cytoplasmic membrane. All right. Gloria, do your cells have a cytoplasmic membrane? Yes, her cells do, right? And so do mine, and so do the rest of yours, right? And we'll talk about the cytoplasmic membrane a little bit. What's really cool is that for the most part, the structure and the function is very, very similar in prokaryotic cells and eukaryotic cells. You see here point number 2, and this is highlighted by the green. This is going to be the cell wall, all right? Now, you guys do not have cell walls in your cells. However, right, some eukaryotic cells do, like those that we find in plants or those that we find in fungi. But remember, and we'll talk about this a little bit more when we get to it, but the components of cell walls between prokaryotic cells and eukaryotic cells are different if they are even present in those eukaryotic cells. And then last but not least, right, we have this layer called a capsule, all right? And a capsule, we'll talk about, is kind of like this slimy, sticky layer that helps cover bacteria, right? All prokaryotic cells, and if you wanted to Google me and challenge me on this, you totally could, right? All is a dangerous word in biology because there are always exceptions. I will point out one of those exceptions when it comes to a cell wall and bacteria a little bit later. Right, disclaimer, when I say all, let's go with Dr. Dactus saying 99%-ish. All right, I'm trying to teach you some of the general principles. I will point out exceptions. So just kind of keep that in mind for anything moving forward for the rest of your life when it comes to science. All is a dangerous word, right? But in this case, all or most bacteria are going to have a cytoplasmic membrane and a cell wall, all right? And we see capsules in some. If we were together, we would actually be doing multiple stains that would look at the structure of the cell wall and would look for the presence of a capsule. Unfortunately, we will not be doing that, but I do have some videos and some activities that you guys will be doing that will cover some of these stains. All right. So let's kind of talk about these components of the cell envelope. And we'll start with the cytoplasmic membrane. Hopefully this looks somewhat familiar to you guys, those of you that have had ANP1 and Bio 2. If you fall into either one of those categories, have probably seen this phospholipid bilayer, right? And what's cool about the phospholipid bilayer is that you see a hydrophilic component right, on each side with a hydrophobic component in the middle. And this is really important for keeping things in and keeping things out. All right. Remember, phospholipids are a type of fat, right, that has been modified. And these unique principles, all right, create, right, what we call a selective membrane. All right. We'll talk about some things, and I think you guys actually did this on your little mastering assignment, right? Some things freely flow across the cytoplasmic membrane, right? Some things do not. Before we jump to the next slide, we can pull this forward. Maybe we can pull this over here. Let's see, I'll pick on, who should I pick on? Olivia, for things that can't get across, and we're going to talk about this in just a minute, for things that can't just cross this membrane, how do they get to the inside of the cell? Or how do they affect the inside of the cell? What can we add to this membrane?
I honestly have no idea.
No idea? Okay, who can help Olivia out? That's fine.
So like a protein, like a not transport protein, but
Well, you can say you can say
I mean,
transport protein.
that's like the active, like
Right.
with energy, but a gated channel protein, like not gated channel protein.
No, this was one of the extra credit questions on the quiz game that we played together. I'm gonna have to go take a point off from your score. I'm just kidding. So Noelle's right. So transport protein, right? If we wanted to be really general, we could say a transmembrane protein, right? And transmembrane proteins are able to span, all right, the outside to the inside of a cell. And Noelle's right. Some of them, right, transport things. Some transmembrane proteins do. Some transmembrane proteins are simply receptors that pass a signal along without something necessarily getting in. All right. Good. All right. So look, there it was, one slide ahead, right? So Noah's right. So transmembrane proteins, they can be gates, allow nutrients in, allow waste out. We will actually see some bacteria, some drugs that we give bacteria actually try to enter through these transmembrane proteins. And so one way that bacteria develop antibiotic resistance, or that they are resistant to antibiotics, is they can actually close these channels in the presence of some of the drugs that we're using. Some bacteria can also, once a drug is inside the cell, can pump it out, right? And so this is unique genetic information that these bacteria have, and sometimes they share this information with their friends, and that's how we can get a lot of drug-resistant bacteria when we didn't have a lot to begin with, and we'll go over that. So selectively permeable, that's what I'm saying. So some of these, what are these things that can easily pass? Oxygen, nitrogen, carbon dioxide, water, and other small hydrophobic molecules can cross. And one of the things that hopefully you can see on some of these is that some of these are either elements or molecules that are just so essential to life that it makes sense that they can pass through. All right, so like oxygen and water are just hugely important in a lot of chemical reactions. CO2 is a common waste product that's generated during metabolic activity, and so it makes sense that it can easily cross, and typically CO2 is crossing on its way out. All right, again, some exceptions, but typically crossing on its way out. And so you just want to allow these molecules and atoms to move freely so that you're not expending energy to transport them. All right. Oh, and this is this idea of these are moving via simple diffusion. Tumi, can you remind me really quickly what diffusion is?
It's like passing from like more concentrated to less concentrated.
Perfect.
Via semi-permeable membrane.
Right, perfect, right? So you have a high concentration up here, you have a low concentration up here. right, that membrane is permeable to these couple things I presented to you, so those things are going to want to rush that way. Okay. So what's really cool here, right, is this idea of osmosis. Okay, and in this case, you know, what we see in a cell wall that we don't have in our cells is that the cell wall actually allows, all right, or provides some support to an expanding cytoplasmic membrane. So if you remember the basic principle of osmosis, osmosis essentially is the diffusion of water, right? When we talk about movement of water, we're talking about osmosis. So here, right, let's say this is the inside of the cell, here's the outside of the cell. You can see a hypotonic solution, right? That means a low solute. For those of you that always forget maybe what solute, think of solute as like a salt or a sugar, all right? So here you have a low concentration of this solute, so a lot of water. Things like to be balanced, right? We like equilibrium, homeostasis, and so water flows in to help dilute this internal solute concentration, okay? At the same time, right, or we'll say a kind of the opposite situation, the solute concentration on the inside is low while it's high in a hypertonic solution on the outside, and so water flows out. And you can see the different effects that it has on these two prokaryotic cells down here, all right? So as water flows in, you see the cytoplasmic membrane pressed all the way up against the cell wall, right? The cell wall actually allows, right, a little bit of extra expansion, right? It provides support. Think about like a pipe or pressure, right? You put a lot of pressure on it, eventually it might burst. That cell wall provides a little extra time, right? Some extra fluid that the cell can take in. And you can note here, right, if we start to pump a bunch of water out to dilute a surrounding environment, it will become, all right, it'll shrink, right? It'll become much smaller. It'll pull away from that cell wall. This is, you know, why, you know, if you would take a bunch of bacteria and just toss it into a bunch of salt water, right? This is the effect that you would see, all right? And cells can survive these conditions for a while, but for the most part, they're looking for equilibrium. Now, one of the things that this doesn't show, and I wish they would show this more with osmosis, is at the same time water's flowing in, you more than likely have a pump, right? If these things can't easily pass, or right, if they're easily permeable, you might also have the flow of solute going out too, right? So it's the movement of water and the movement of solutes that are trying to form these osmotic conditions, all right? And so this is a reason if you ever do research with any type of cell, you can't just put them in regular water. You can't just put them in plain salt water, right? You have to be very, very careful with the concentration of the solute in the solution that you put those cells in, because you could cause them to shrivel up and die, or you could cause them to swell up and burst, right? So they're looking for this balance. And again, so far, a lot of these principles almost exactly identical, right, between prokaryotic cells and eukaryotic cells. All right. So again, this is review for a lot of you, but I still think it's important concepts to reinforce, right? So we've talked about simple diffusion, right? Passive transport. So Toomey said right here, you have a high concentration, here's a low concentration. So these little blue guys are going to want to rush in. You also have facilitated diffusion, all right? And so this is kind of what Noel was referring to with a transmembrane protein. In this case, right, it simply serves as a channel for these little triangles to pass through. Now, it's important to note that in both prokaryotic cells and eukaryotic cells, these channels are selective, all right? So if we're going along with the theme of this picture, right, what you don't have is you don't have these little blue circles or these little green squares entering through this same channel, right? This channel, although it does not require energy, right? Diffusion doesn't require energy. It's only letting these triangles in, right? In this case, maybe a certain nutrient or certain molecule, right? Active transport, right, requires energy, right? And it requires energy because you're going against that concentration gradient, right? So here you have a little, here you have a lot. And so normally you would think, well, okay, the lot's right here, and then it goes, right? It would want to leave the cell. And that may be true, that may be what it wants to do, but this might be a really important nutrient. And so we're trying to bring in all of it that we can, right? Or the cell's trying to bring in all of it that it can. And so, right, you're going against a concentration gradient, which requires energy. Now, one thing that I kind of wanted to talk about that I think is really cool, and as you guys go on through this course, you may come across the term exoenzyme, right? And an exoenzyme is something, right? An enzyme that leaves a cell and does something outside of the cell. Let's see, I'm trying to dig back. Who had, we'll go with, hey, Allie, do you remember if a eukaryotic cell is trying to engulf or take in a large molecule, do you remember what that process is called?
Endocytosis.
Yes, endocytosis. Good, right? So, and we'll talk about this briefly in the eukaryotic portion of this lecture if we get to it. But endocytosis, all right, is basically you engulf and you take your membrane and you surround a big thing, bring it into the cell, right? And it becomes something called a phagosome. And again, I'll show you some images in a second. And then the big difference is digestion takes place within that cell, all right? Prokaryotic cells, all right, have cell walls. Things with cell walls, right, it is much more difficult, right? Very complicated. They can't really do endocytosis, right? They can't bring in things that way. There are some cool, I actually have a paper on it. It's not the most interesting read in the world, but scientists have found kind of a bacteria that's kind of a, we'll say a tweener, right? It is bacteria, but it is capable of a modified, simplified version of endocytosis, right? But for the most part, we say bacteria aren't bringing in things via endocytosis. What they are doing is they are using these transmembrane proteins that are embedded within the cytoplasmic membrane, all right, to send enzymes out, okay, to digest macromolecules, right? So a macromolecule, remember macro just means big. So this is a big, you think about it as a big complex sugar, maybe or something like that, right? Breaks it down into small subunits that can then be taken in, right, via other transport proteins where those nutrients can be utilized by the cell. All right. So again, this is just for the big macromolecules. The smaller molecules, right, really tiny nutrients, some of those can just cross the membrane and be utilized, right? But if we're talking about something that's really big that needs to be broken down, a bacteria is going to need to release an enzyme, right, in order for that to happen. And just, you know, one other thing here to point out is you can see, right, this kind of pre-protein with a signal sequence. The signal sequence is what allows this enzyme or tells this enzyme to leave the cell. And we call it a pre-protein because it doesn't fold until it leaves, right? So if it folded before it left, it wouldn't be able to cross. All right, so once it gets outside of the cell, okay, it will form an enzyme and break these down. Now, what this picture doesn't show, and you'll see this here in just a minute when we get to the cell wall, is that this is not the void of space out here, right? What you'll see in just a minute is that the cell wall is somewhere kind of up here, right? So these enzymes aren't just floating around freely, right? The cell kind of still keeps them in a vicinity that it can utilize them. And you'll see what that looks like in just a second, right? You guys will also see a little bit later that fungi do or have a similar way of digesting large macromolecules. So in that case, right, their way of taking in nutrients is kind of similar to prokaryotic cells. Ours is very different. All right. So let's talk about energy. Lori, can you remind me from yesterday what membrane-bound organelle in eukaryotic cells generates energy?
Mitochondria.
Yeah, the mitochondria does. And so when Glory said that, I said, that's totally right. But what's really cool about prokaryotic cells is that the whole cell kind of acts like a mitochondria. So if you talk about a eukaryotic cell, although people that do mitochondrial research for some reason get deeply offended by this, right? I consider the mitochondria a battery, all right? It provides energy and power to the cell. For a prokaryotic cell, for a bacteria, all right, or an archaea, right? The whole cell is a battery. So one of the things, right, that I hope you remember is that the electron transport chain is embedded within the mitochondria of a eukaryotic cell, all right? And you have different proteins, different transporters that are moving things around, right, to generate a proton gradient, right? The whole membrane of a bacteria, right, embedded within it is going to have these electron transport proteins and carriers, okay? And what we'll also talk about a little bit later is this proton gradient that's used in your cells to make energy. It's used in prokaryotic cells to make energy, but it also has some other unique functions, all right, that we see in prokaryotic cells that we don't carry out, all right? So we'll go over that more, but this is important, right? The mitochondria is a battery. The whole bacteria is a battery, okay? And again, we'll kind of revisit that because that's important for some evolutionary principles about how things came about, right? So let's talk briefly about cell walls, right? Gram-positive and gram-negative bacteria. So this is a cell wall. Pretty cool, huh? So notice down here, this is the cytoplasmic membrane that we just talked about. So remember, we were talking about those exoenzymes, those enzymes that are pumped out of the cell, right? And so they're going to be in this area, pulling in nutrients that will then enter back through the cytoplasmic membrane, right? So this enzyme is not just getting pumped out into the abyss, right? It's kept, right, within, essentially within the cell, okay? So for the cell wall, you have two important components. N-acetylglucosamine, also called NAG, right? And NAM, right? N-acetyl-muramic acid, all right? I'm fine if you refer to them as NAG and NAM. You can see here they're making the purple NAG and the blue NAM, all right? So you have alternating chains of these, right? Alternating peptides. And then you can see that these sheets are then stacked on top of one another. Now, what you're looking at right here is a gram-positive cell. The structure of a gram-negative is slightly different, and we'll go over that in just a second. What I want you to know is that in the sheets that are laid on top of each other, right, holding those sheets together, right, are tetrapeptide chains that connect a NAM molecule above to a NAM molecule below. And in gram-negative bacteria, you have a direct linkage between NAMs that are on parallel sheets. And in a gram-positive, you have this intrapeptide bridge, right? And I'll show you kind of zoomed in what that looks like in just a second. Now, hopefully this makes sense to you, right? This cell wall is permeable to sugars, amino acids, and other nutrients. And hopefully that's kind of clicking. That makes sense because they have to cross the cytoplasmic membrane. So if your nutrients can't get past the cell wall, well, then they can't get to the bacteria and the bacteria dies. All right. So this is also a permeable layer, right? Or at least we'd say semi-permeable layer. Are we good here? You got that, Glory? Yeah? All right. So this is, right, the link between two parallel strands of peptidoglycan in a gram-positive cell, all right? So here you see gram-positive has this peptide intrabridge, right, that connects these tetrapeptide chains. You can see that. So you kind of get this, I don't know, I guess I kind of call it, it looks like a chair to me, right? Here's the back of the chair with a little bit of a leg, right? Here's the seat and here's the rest of the leg, right? I realize it's not an even chair, but I feel like that's kind of a good way to describe the general structure of this, right? In a gram-negative cell, right, what we see is that you have a direct linkage, right? This tetrapeptide bridge is going to run all the way down to a NAM molecule on the parallel strand, right? So gram-negative, right, they lack this peptide intrabridge. Okay. All right. So let's talk about some of these different components of a gram-positive cell wall. And, you know, as I point these things out, right, just do your best to kind of pay attention and maybe get some of this down, because what you will see in just a minute is that the cell wall of a gram-negative cell is very, very different than that of a gram-positive, right? Kind of the general structure. And I know it maybe seem confusing now, but as soon as you see it, you'll be able to notice these differences. And what I would suggest while you're studying and preparing is to get an image of the gram-positive cell wall and the gram-negative cell wall and put them side by side. And those differences should jump out. Right? So one of the things that we will see and discuss is the fact that this peptidoglycan layer in gram-positive cells is very, very thick, right? And you'll see the difference between that and gram-positive in just a second, right? But you have a lot of layers stacked on top of each other for this gram-positive cell wall, right? You also see here the presence of something called teichoic acid, all right? Teichoic acid, you know, has a couple functions. There's still a lot that we don't understand about it. We do, right, know or think at least that it helps stabilize the cell wall. And they also, right, can basically be ion sponges. And so one of the things that we don't really get into a lot in this class is the fact that for some enzymes to function properly, they need something called a cofactor, and a cofactor can be an ion. And so that ion helps activate an enzyme, and then it can do its job, right? And so if you're pumping enzymes out, right, into this gel-like space, you might need certain ions to help that enzyme achieve its functional capabilities, right? So they provide stabilization, and they also are attracting some ions, all right? So between, right, again, the cytoplasmic membrane and the cell wall is this gel-like layer. So remember, those enzymes that are getting pumped out, right, are not just disappearing, right? They're not just going away. They're staying near the cell. Nutrients are coming in because they can pass through the cell wall, right? So then we can digest, or some of those nutrients, if they're small enough, can directly cross the cytoplasmic membrane. Okay. And we'll talk about in lab, or some of the videos that you'll watch in lab, we'll kind of address the principles of how we use the cell wall to our advantage when we're staining bacteria. Now, this is the gram-negative. I'm hoping that some things kind of immediately jump out to you, right, as being different, right? And so I'd say kind of the two major differences, and we'll talk about some components that then go with these differences, is that here, all right, you see a peptidoglycan layer, but it is really thin, right? So it's a thin layer of peptidoglycan. I don't think they do a great job here on this drawing. They kind of make it look like the intrabridge. That shouldn't be there, right? There should be more of a direct kind of linkage there, but that's okay. And then you see this whole other membrane, all right? So gram-negative cells actually have two membranes. They have the cytoplasmic membrane, right? And then they have an outer membrane, right? And we'll talk about these in a little bit. Well, we'll talk about them more here in just a little bit, but we'll also talk about them in antibiotic resistance chapter. But these are poron proteins, right? So think about a pore, a hole, or an opening, right, that are in this outer membrane. And hopefully that kind of makes sense to you guys, because remember, not everything can cross a membrane. Now you have an extra membrane. And so you need a way, right, for nutrients, all right, to cross into this periplasmic space, right? So the space between the two membranes that has the cell wall embedded within it, right? You need nutrients to be able to cross. And so to cross this outer membrane, some things are going to need an extra channel, right? What you also will notice here, right, is something called LPS, lipopolysaccharide, right? Also called endotoxin. Does anyone remember one of the questions that you answered last night that was related to endotoxin or LPS? Does anyone remember going over that?
Was
Meg,
it that?
No, go ahead, Noel. If you remember, do it.
Was it that it like caused inflammation? Oh, I don't.
Right. So let's go. Right. So the question,
Yeah.
right? LPS is great at triggering fevers, and so the question, had something to do with you have a patient that's sick and they start taking antibiotics, and it causes a small spike in their temperature, right? And the spike is because it's killing all these gram-negative bacteria, right? And then LPS is getting released and causing an inflammatory response. All right. So LPS is kind of a big deal, right? So for LPS, right, lipopolysaccharide, also called endotoxin, right? So you have the O antigen, right? That's that outer portion that kind of looked like the red hairy stuff, which is variable amongst bacteria, right? So although LPS is similar, there are varieties within bacteria. And then you have lipid A, right, which is the portion of that molecule that's anchored in the outer membrane. This is the part that is highly inflammatory, right? And so when a cell bursts or when a cell is killed, sometimes, right, this lipid A gets released and it causes an inflammatory response. Interestingly enough, some researchers, or what we've seen is that a Western diet, so when we eat a lot of things that we like to eat here in the good old US of A, actually causes a disproportion of gram-negative and gram-positive. So you have more gram-negative bacteria. More gram-negative bacteria means more LPS. And what we think is that part of the problems with obesity And this bad diet is that you're constantly having LPS stream into your circulation. So it actually induces a chronic low-grade state of inflammation, which then increases the risk of metabolic disorders and cancers. All right. So, yeah, I don't know. It's just kind of cool to think that all of that stuff can happen, right, because of LPS on this outer membrane. All right. And so this is just kind of like a little, side note, but if any of you ever go into any type of research or work in a lab, LPS is really good at activating the immune system. And so if we're studying cells, even if they're in culture, sometimes what we'll do is we'll put a little LPS on them to activate them and kind of study what they're like in that activated state. This is the first time that I want to introduce something called a pathogen-associated molecular pattern, all right, a PAMP. LPS is one of multiple PAMPs, all right, that could be within your body, all right? Pathogen-associated molecular pattern. What that means is that your body is looking for patterns, all right, that it knows are not part of you, all right? We'll talk about the innate immune system, all right? The innate immune system is looking for patterns. It's looking for things they can easily flag and say, that is not part of you. That's probably part of a pathogen, all right? So a PAMP is a pattern that your body recognizes, right, as belonging to something foreign. It then starts to generate a response, okay? Now, this usually breaks my heart when I introduce this because I always ask for PAMPs on exams, examples of a PAMP or what they do, and inevitably someone puts down TLR4. TLR4 belongs to a group, right? And write this down, don't make me sad, of something called a pattern recognition receptor. Pattern recognition receptors recognize patterns on PAMPs, all right? So pattern recognition receptor is how your body is recognizing the presence of something foreign. So in this case, LPS is a PAMP that is recognized by TLR4, which is a pattern recognition receptor. All right?
Can you say that again really quickly?
That.
Just the last part of how you.
Yes. The last part. Okay, so LPS is a PAMP, right? It's a pathogen-associated molecular pattern. It is recognized by TLR4, which is on your cells, and TLR4 is a pattern recognition receptor. Does that make sense, Laura?
Yes, thank you.
And I will point out, even for like this part of, this unit, right, between now and I guess tomorrow, I'll point out another really important PAMP that your body recognizes as belonging to something foreign. All right. So I mentioned, and I'm just going to go for a few minutes and then we're going to take a little break because this is a lot of information. So gram negative bacteria, because they have that outer membrane, right? You saw those little yellow things that I said were porons, right? That's because we need nutrients to get past that outer membrane, right? Gram positive just have the cell wall, which is permeable to almost all of the nutrients and the good things that a bacteria needs. And so we don't see them there, right? So porons are present in gram-negative bacteria only. We don't see them in gram-positive. Now, this is one of the things that I mentioned. Please don't think that a poron is just a big opening that allows things to pass that outer membrane. All right? These are very specific, right? They can use them to move things in and move things out, and bacteria control them, can control them to the extent of not allowing or helping to push things out like antibiotics and maybe trying to harm them. One of the things that we will kind of discuss when we get to the antibiotic portion of this class is the fact that a lot of time going after gram-negative bacteria is a little more tricky because they have some kind of built-in defense mechanisms. All right. So the periplasm, right, is only found in gram-negative bacteria. However, I do want you to note that it is very similar to that gel-like layer if you go back and look at the gram-positive cells that exist between the cytoplasmic membrane and the cell wall. All right. But within this periplasmic space, and again, you're going to have similar things taking place in that gel-like layer in gram-positive. This is where nutrients are broken down. Remember, they're secreting exoenzymes. All right. And so these two kind of go together. Electron transport, right, is taking place in this space. And so this is where that proton gradient is helping to be established. But remember, those components, those proteins are embedded within the cytoplasmic membrane. And then here you have something called xenobiotic metabolism. And basically, this periplasmic space is a great space. And the same principle applies to the gel-like layer of a gram-positive cell. If you can break down a drug or neutralize some other threat that's in that space, you're actually going to be fine as long as you prevent it from getting to the cytoplasmic membrane. So that's what's going on here, right? This space, this periplasmic space here, or this gel-like space in the gram-positive, gives you an opportunity to neutralize a threat before it can interact with the cytoplasmic membrane, right? That is usually when destruction of a cell occurs, right? Is when that cytoplasmic membrane is being altered or affected. This is for all of my nerd friends out there. What's the price now? I haven't checked it recently. This price is a little old. Let's see. Yes, this is on Amazon. It's a textbook just on the periplasm, right? So if you ever are getting bored in microbiology, if you're ever like, why are we doing this? Remember, it could be a lot worse, right? You could have a textbook on just the periplasm. If you like it though, kudos to you, right? Go get it. Let's see. Yeah. Okay. So this is where we're actually going to take a break. It's like 9:05. Let's just take like a 10-minute break. I'm just going to get up and walk around. I'm going to leave the Zoom page up. All right. So you don't have to sign off if you don't want to. If you do, you can sign back on. We'll be here, but I got to get up and stretch, and we've been talking for like an hour, and that's a long time. All right. So this is a diagram that I find really useful, and in your textbook you'll find something similar, maybe a little different, but I like it because it just kind of lays out, you know, some important structural components of gram-positive, some important structural components of gram-negative, and puts them side by side. Now, one thing I do want to point out on this chart is the fact that I totally agree. Cells that stain purple are more than likely gram-positive. Cells that stain pink, and you can see these over here on the side, are more than likely gram-negative. On an exam, though, telling me that one is pink and one is purple is not going to get you credit, right? And the reason they're pink and purple, and again, we'll talk about this maybe a little bit more in lab, is the fact that the gram-positive has a thick peptidoglycan layer compared to the negative layer, or I'm sorry, to the thin layer on the gram-negative cells, right? And then also some of the treatments affect the outer membrane and cytoplasmic membrane, where they only affect the cytoplasmic membrane in the gram-positive, right? So that's what's contributing to the color. And so you telling me that one's purple and one's pink as part of an answer doesn't really show me that you're kind of understanding some of those differences. Now, one thing I want to point out here, and I have this on the next slide too, is the sensitivity to penicillin and sensitivity to lysozyme. All right. And so gram-positive are going to be sensitive to penicillin and to lysozyme. Lysozyme essentially is a natural antibiotic that's produced by your body. There's lysozyme in your tears. Your gut cells produce lysozyme. Lysozyme was discovered by Alexander Fleming, who also discovered penicillin. And so the two have an interesting link there. Gram-negative, primarily because of this outer membrane and some other features, are typically resistant to both of these. Now, very briefly, and we'll talk about this more in the antibiotic unit, is these molecules are functioning, quite differently. All right. So first of all, right, penicillin is actually a fungal product. All right. It was discovered, I believe Fleming was trying to grow some Staphylococcus aureus on a plate. The plates became contaminated, and he noticed that there was kind of a zone of separation between the fungi and the bacteria, right? And so that's how he started to kind of say, hey, maybe this is something important here. It inhibits the linkage of peptidoglycan, all right? And what this is a fancy way of saying is that when a cell is dividing, right, and you're making another cell, you're putting together those peptidoglycan layers. And that's when this antibiotic is effective. And so if the cell is not dividing, then penicillin is not going to be effective at interrupting cell wall synthesis. All right. And basically what this does is it forces the cell to constantly be remodeling, and the bacteria actually ends up tearing down its own cell wall. And once that cell wall is down, then the bacteria can become very susceptible to other medications or the immune system. For lysozyme, what's interesting about it is that it is breaking the linkages between NAM and NAG. So it doesn't have to be building, right? The bacterial prokaryotic cell can just be sitting there, and NAG and lysozyme will come in and start breaking those bonds apart. And again, typically more effective against gram-positive, not because this wouldn't work on the NAG-NAB linkages in gram-negative, but it's not able or does not have as easy access to that cytoplasmic membrane because of that to that cell wall because of the outer membrane. All right. And again, we'll talk about those more a little bit later. Now, you do have, remember I pointed out some interesting differences. And so mycoplasms and mycobacteria actually lack a cell wall, all right? And so this makes them very difficult to kill. So again, please don't overthink this. Caroline, why would lacking a cell wall make it difficult to kill with a lot of our antibiotics?
Well, if like you were saying, they break apart the NAG-NAMs, then if it doesn't have that, then it's already there's nothing to break apart.
Right. So perfect. What you'll see when we get to the antibiotic chapter is that we have a lot of variety of antibiotics, but we are still heavily reliant on antibiotics that we have derived from the penicillin family. All right. And so most of the things we've derived from the penicillin family are looking specifically at targeting cell walls. If you don't have a cell wall, right, it is very, very hard to target with that antibiotic, right? So they really won't work. Archaea bacteria, right? I just kind of, I kind of put these in here because their cell wall is much more diverse even amongst different archaea bacteria. So we have a general uniformity within prokaryotic, right, bacteria. We see a different kind of cell wall structure in archaea bacteria. They lack peptidoglycan, but they do have a peptidoglycan-like molecule that essentially forms a almost identical structure. So let's talk about mycoplasms and mycobacterium really briefly. Up to this point, we would consider most of these to be parasitic. And, you know, by this, what I mean is that in some cases, even bacteria that may cause illness or other bacteria that may cause illness in you, we kind of have a good idea of what its role is or its niche in the environment, right? And sometimes it just causes disease because you come across and interact with a part of an environment that you don't normally do. As of somewhat recently, and I looked this up maybe a couple of years ago, so again, things change, but we don't really know the natural role or the natural niche of mycoplasms, right? Again, that could have changed recently as we continue to learn more and understand. But what we do know about these type of bacteria is that they like to adhere tightly to epithelial cells, right? And that's going to be the cells that are in you. All right. So those of you that have had ANP, remember you have epithelial cells everywhere, right? In this case, right, you do have some in your lungs. You do have epithelial cells in your urogenital tract, right? And so these cells are going to, or these bacteria are going to adhere tightly to these cells, and they're going to release something called hydrogen peroxide, right? And hydrogen peroxide, maybe you've used that like when you have a cut or a wound. And hydrogen peroxide is something called a reactive oxygen species, and it can have very negative effects in large quantities on your cells, right? So this can cause a lot of damage. What's really cool about these bacteria is that they have a high requirement for cholesterol. So, you know, I kind of think cholesterol gets a little bit of maybe a bad name because we're always worried about cholesterol levels. How high is that food in cholesterol? And it's true, just like anything else, too much of it in your system can be bad. But cholesterol, even in your cells, actually plays a really important role in providing structural rigidity to your cytoplasmic membrane, all right? So we don't have a cell wall. So we need something to essentially give a little more strength to our cytoplasmic membrane. And cholesterol actually does that for us. And so what we see in some of these mycoplasma and mycobacterium is that they have a very high level of cholesterol within their cytoplasmic membrane, to the point of it, that cytoplasmic membrane actually becomes much more like a cell wall. All right. If we were to grow some of these bacteria in lab, you kind of have this egg-like look where the center is raised, right, like an egg yolk, and then the egg white, right, lays a little flatter, closer to your plate or closer to the bacterial plate if we were growing them on them. You also have an interesting group of these. Again, I mentioned they were present in the urogenital tract, and they actually use urea, right? And urea, right, is an important component in urine, right? So it's kind of like just a great nutritional source for them if they're present. Some mycobacterium and mycoplasm, right? Mycoplasma pneumoniae, Mycobacterium tuberculosis, right? You know, these associated with bad lung infections. What's really interesting about these mycoplasms and mycobacterium is that they divide very, very slowly. So another thing that antibiotics are trying to kind of take advantage of, in addition to cell walls, is that a lot of them are basically targeting things that are dividing at a very, very rapid and quick rate. All right. Bacteria divide typically much more quickly than your cells. And so medications are going to affect them more drastically. A similar concept to chemotherapy, right? Chemotherapy, if you've known anyone that has cancer or has even been treated by radiation, the idea is that cells that divide rapidly, all right, are more affected by that treatment. And that's what we try to do with our antibiotics. It becomes really tricky and challenging because these bacteria divide very slowly. All right. So just kind of a cool, unique group. And we may mention those a little bit later if we talk about some pathologies associated with them. Archaeobacteria, again, I kind of just end up grouping these with, prokaryotic cells. And in the next chapter that we'll get to here for a little bit, we are going to talk about extremophiles, which a lot of times are archaeobacteria. Some bacteria fall into that category too. But we don't really give these guys maybe enough attention. There's still a lot of research that needs to be done on them. Researchers are very confident that archaeobacteria are even present in your gut, but what their roles and what they're doing, right, we still don't know, right? And these were discovered, again, remember I mentioned we thought they were bacteria when we did analysis of ribosomal RNA. And we'll talk about that a little bit more. This is a really powerful tool in grouping and categorizing bacteria. And we were looking at methanogenic bacteria that are capable of producing methane gas. And we noticed that when we were doing genetic analysis that these looked significantly different on a genetic level from bacteria, right? So some of the major groups are methanogens, extreme, halophiles. Rachel, Dickey, do you remember what a halophile is? Did you guys have to look that up yesterday in lab?
Yeah, was it like really extreme? I don't remember which one it was.
This is really extreme something. Can someone help her out? Does anyone remember?
Is it they need a high salt environment?
Right. So a halophile is referring to something that can survive in a high salt environment. All right. And then thermoacidophiles, and talking about that, high temperature, high acidity. All right. And in this case, what's even more interesting, right? Not only is their cell wall slightly different, their cell membrane actually has a different branching pattern in the hydrocarbon chains of those phospholipids, right? And so, you notice some of these actual physical differences, and then makes sense when you analyze the genetics and say, hey, these are actually different. All right. So, that was kind of a long and roundabout way. We did the cytoplasmic membrane, we did the cell wall, and now we're going to talk about the cell envelope, or the rest of the cell envelope, right, which is going to be the capsule. So remember, not all bacteria have capsules, but some certainly are capable of forming these, right? So, capsules are important for a couple of things. What can you guys imagine capsules may be important for in a bacteria? You can use maybe the image below to maybe start helping a little bit. But what could you see a capsule being beneficial for or important for? Lauren, you got anything? Oh, go ahead. Oh, Rachel, do it. Nope, you got one. I'm going to get the other one for Lauren.
To stick to something?
Okay, so sticking, right? Perfect. Attachment. Capsules are really important for allowing attachment. Awesome. What else? Lauren, you got anything else for me?
Well, it says protection, so I'm going to say probably protection.
Very good. I'll go with protection then, right? So drugs, antibodies, that we'll talk about more, things that are trying to attack the bacteria can sometimes be stuck, right? Get stuck in the capsule. If you're stuck in the capsule and you can't interact with the cell wall or the cytoplasmic membrane, right, then in essence, you've been neutralized, right? Or, right, that capsule makes it hard for an immune cell to interact with the bacteria. If it can't interact, it's much more difficult to kill it, right? So attachment, right, bingo, right, and protection. Slime layers and capsules. And so what's the kind of difference? On a molecular level, they're really similar. A capsule is going to be more uniform, right? While a slime layer is going to be kind of more slimy, right? And so here what you actually see the formation of is a biofilm. This is actually Staph aureus, right? So when you have this slime layer and these encapsulated cells, again, that makes it much harder to treat these infections. And you can imagine if you've known someone, that's had surgery and they get like an S. aureus, right, or like a MRSA, right, which is methicillin-resistant Staph aureus infection in their bone, right? This can be very hard to treat. Those of you that go to the dentist regularly, which I would recommend, right? Dental plaque, right, is formed by something called Streptococcus mutans, which I think you guys had a question on yesterday. And so although this is a different bacteria, this is kind of what a dental plaque looks like on your teeth, right, if you don't take care of them. All right, and we'll talk about those more later. Now, kind of associated with that outer part of the cell envelope, and you'll see that there's some different anchor points for this, though, are flagella, right? And flagella is a major mechanism of bacterial propulsion. And as you can see in the images on this slide, that you can have one flagella or you can have a lot of flagella. And you'll see here on a diagram, and you even answered a question about this yesterday, that they have an anchor point in the cytoplasm and the cell wall, all right? And if you think about it, right, this is a motor that's generating force, and so it needs to have a strong anchor point, or else the motor will come off, right, and the bacteria will be left there just floating. What's really cool about pathogenic bacteria is that we know that their flagella are more powerful, and these powerful flagella allow them to penetrate mucous membranes. And so on your epithelial cells, or a lot of your epithelial cells within your digestive, respiratory, and urogenital tract, you have a mucous membrane. And this mucous membrane that lays over our epithelial cells kind of acts like a capsule that we see in bacteria. It provides protection to those cells that are below it. Things get stuck in it. A lot of bacteria get stuck in it. We then move that mucus out, and you never have to worry about it. Bacteria that are pathogenic, right, are strong enough to swim through that mucus where they can come in contact with your epithelial cells and then cause the damage that we associate with disease. Right. So this is what a flagella looks like in a gram-negative bacteria, right? Allie, how can I tell that this is a gram-negative bacteria by looking at it?
In the picture, you see the two different membranes and just all those little structures that are part of the gram-negative.
Right, I would do, right, you have the two membranes, and then you have a very thin layer of peptidoglycan, right? So that should be an immediate ding, ding, ding that that is a gram-negative bacteria. So the flagellum can be divided into three basic components, right? It can be divided into the filament, right? So this is the part that's actually, that you see kind of flipping or whipping, right? You have the hook, and the hook is a connection point between the filament and the basal body. And the basal body, right, is what's being anchored here, right? You see in the cytoplasmic membrane and in the cell wall, all right? And so this is going to provide propulsion. I just wanted to point out another thing here to make you guys start to think about this. So here you see proton pumps, right? And so these are the proton pumps that are embedded within the cytoplasmic membrane that are important for electron transport, which generates energy. But in this case, what we also see is that proton gradient that in our cells is used to generate energy, and in prokaryotic cells, that proton gradient here is also used to actually provide the propulsion that allows the flagella to spin, all right? And this is just kind of a diagram. So those are protons, right? Protons are positive charge. They move in. When they move in, right, they cause a spinning motion, which causes the flagella to have its whip-like motion, which allows propulsion of the bacteria. Now, what does a bacteria propelling itself look like? All right. And to describe that, right, we have this run-tumble paradigm, all right? So before we talk about this motion, right, what causes a bacteria to move, right? And what causes a bacteria to move, right, is its ability to sense chemicals or nutrients, all right? Now, what I have here is I say here's a lower concentration, here's a higher concentration, right? So typically, if it's a nutrient or something good, the bacteria is going to move towards the higher concentration, right? That's where the food is. What I don't have on here, though, some bacteria actually have the ability to do the opposite, right? They can sense that this is a bad chemical, right, that's present, right, or a bad molecule. And so they will actually move towards the lower concentration, right? So it depends. Is it moving to eat or is it moving to escape, right? They can use that concentration gradient. So this is what we call chemotaxis, all right? So it's the movement, right, by sensing a certain chemical or nutrient, right? The way I kind of, describe this motion right here, you see the little X, this is the target, this is where it starts. Bacteria do not take clear paths. In fact, right, when a flagella, right, is propelling a bacteria, it actually spins in a counterclockwise motion. And eventually, right, it spins so much, right, that it can't spin anymore, right? It basically tightens up. And then it has to unspin, right, or spin the opposite direction to kind of relax it, right? So if you've ever, I don't know if I have it, let me see, hold on. So if we're in class, I don't know if this will work quite as well, but this is kind of the representation that I would give of, this is my bag, right? I spin it. So think about this spinning in a counterclockwise motion, right? Eventually, it's going to get tight. It can't spin anymore, right? I let it go, and it unwinds, all right? And this is what flagella do, and this is why you see the run, tumble, run. So during the run phase, this bacteria is moving in a direction towards its target, but eventually that flagella spins. It can't spin anymore, so it unwinds. So during the unwind portion, right, or the tumble, you kind of have this chaotic movement, right? It can't really control what direction it's going. However, eventually unwinds, and then it can wind back up, right? And then it moves towards its target again. Has to unwind, so you have chaotic movement, moves towards its target, right? And then eventually gets to, right, the promised land, whatever that may be. Or, right, maybe we're running away from something. It just depends what the situation is. Okay. For those of you that are believers, right, I would describe this much like our faith walk, right? Very, very chaotic. We eventually get to the target, but it may not be And it likely will not be a straight path for us. Just to kind of give you an idea, and this is not a diagram that I expect you to be able to draw, right? Or necessarily recreate. But I do want to kind of give this as an example of, what might a bacteria be running to, right? What might be attracting it, right? Or how does it sense what it's running to? And so here's an outer membrane, peptidoglycan, cytoplasmic membrane, right? So here again, we're using a gram-negative bacteria as an example, right? And here we have some proteins and we have some sugars. We have oxygen, right, that are detected by these receptors. And when these molecules, right, or nutrients bind to these receptors, they actually, right, signal, right, those flagellar motors to start working to push this bacteria towards wherever these nutrients are highest, right? These are things that it likes, right? So you have receptors that are embedded within the cytoplasmic membrane, right, that are sending signals that allow, right, this flagella to become active and turn on, right? So again, this kind of helps direct it or give it a purpose or a target or goal. Okay? Another important component, right, on the outside of that cell envelope can be something called pili. And pili are going to be one of the ways that bacteria are adhering to surfaces, all right? If we talk about a pili that is allowing adherence to a specific surface, we call this a fimbriae, all right? For me, I don't mind if you use them well, I wouldn't necessarily say use them interchangeably. I would just say just kind of pick one and stick with it. What we know about pili, and we'll talk about these a little bit later in the summer session, is that they're really important for pathogenesis. And so these pili, right, they kind of look like these long hair-like extensions, are actually capable of interacting with receptors on your cells, right? And so that's how bacteria can target certain cells, as their fimbriae can find a receptor and bind to that receptor, and that becomes kind of that anchor point for attachment. All right, this allows movement on solid surfaces. So if we're talking about some type of liquid medium, right, liquid environment, that's going to be flagella that allow these bacteria to move. Fimbriae, right, on more solid surfaces. Again, this is not quick or rapid movement. If we were in class, I'd use the example of if I put some bacteria with fimbriae on the end of one of those long tables, it would not be at the other end by the end of class, right? This is not quick, rapid movement, but it does allow some movement along with adherence. And I think you guys may have actually had a question on conjugation. I can't remember in the set that I gave you. And so we have a special pili called a fertility pilus or a sex pili. And this actually allows or can allow the exchange of genetic information between two bacteria. So this is actually the equivalent of, or can be the closest thing we have the equivalent of bacteria sex, right? That sharing of that genetic information between two different bacteria, right? And we'll talk about that more. I believe that will be next week is when we get to that. All right. So let's talk about the internal structure of a cell. And we'll see if we can at least get through most of this. All right. So I believe Elena mentioned right, the absence of a nucleus in eukaryotic cells. What we see in prokaryotic cells is a nucleoid, right? And so if I go back here for a second, this is the nucleoid. So this is the bacterial chromosome, right? And I hope you heard that correctly, chromosome, right? They have one chromosome. And it is not contained within a nucleus, right? It has a region of the cell, right? It kind of hangs out here right in the middle. If any of you like donuts, right? This is kind of like the filling in the middle of a Long John donut. Ah, so delicious. Anyway, so you see that kind of hanging out right there. Now, I said one chromosome. It is a circular chromosome. Megan, are your chromosomes circular?
No.
Ah, good. Right answer. That makes me feel better. All right, good. So they have one chromosome. It's circular. It's supercoiled, right? And when I say supercoiled, I mean it's wound really, really tight. One of the things that it's wound around are something called nucleoid-associated proteins. Those of you that are biology savants or biology nerds, right, maybe remember that in eukaryotic cells or in our cells, our DNA is coiled around something called a histone, all right? So, you know, when I say supercoiled, right, maybe you're wondering how supercoiled. I have to go back and look at what the chemical is, but basically you can take a chemical and dump it on bacteria and it causes them to vomit out their chromosome, essentially. And so this is one bacterium of E. coli, and this is its chromosome that's been released out of it, all right? So all of that you see outside of the cell is wrapped up and contained in this area right here, right? So very, very tightly supercoiled, right? Now, another kind of unique characteristic that we see in prokaryotic cells, and this does not always have to be the case, but it is in a lot of them, is they have something present called a plasmid, all right? This is much smaller than a chromosome and can be considered, right, in a typical, normal, safe environment to be accessory, right, or non-essential, right? So the way that I kind of look at this, and I think, what are we all? Yeah, 'cause I don't think Trent's here. Okay, so I'm the only guy, perfect, right? So some of you that have a significant other, and maybe you're going out with them on a fancy dinner date or to some party or something, right? And you accessorize, right? You put on maybe your makeup and your blush stuff and your eye stuff, right? And you do your hair up, you know, nice and clearly, right? I'm not much for accessorizing, right? That's, I mean, and I'm sure you could argue with me in multiple ways, but it's not essential, right? You don't have to have makeup on, earrings in, and your hair done to eat food, right? You don't have to have earrings on to go to a party, right? It's an accessory. Is it nice? Yes. But is it essential? No. And so that's kind of what these are to bacteria. But when we think about them in bacteria, we're saying they're non-essential if the environment is perfect, right? If the temperature's right, if the nutrients are there, there's no threat, right? They're kind of living the dream. Now, we know that's not the case for a lot of bacteria. So if we talk about pathogenic bacteria, they are under constant threat, right? Either from us, other bacteria that may be in the same area produce antibiotics. Fungi are really great at producing antibiotics. Interestingly enough, so are sea sponges, right? Who would have known? And so bacteria are a lot of times in the environment under constant threat. And sometimes these plasmids provide bits of information like antibiotic resistance information that are essential to their survival, right? In a realistic environment. Okay. This, I do kind of want you to look over this. We are going to go over this or visit this in more detail in the chapter about replication, transcription, translation. But I still think it's kind of an important, thing to take a little bit of time for you guys to look over. And we've already mentioned some of this: prokaryotic, circular, single chromosome; eukaryotic, multiple, linear. you see some kind of other key differences. I do want you guys to look over these. Again, we will kind of visit these again in just a little bit, though. All right. Another important component that we see here are prokaryotic ribosomes. So prokaryotic ribosomes are composed of two subunits, a 50S subunit and a 30S subunit. You could actually break those down even further if you wanted to, but in this case, we're not gonna worry about that too much, right? And I want you to note that when they're combined, we consider this a 70S subunit, right? And I understand the math doesn't add up. This is actually referring to basically sedimentation time. So when you spin something really, really fast, the heavier stuff gets pulled down more quickly, and you can actually measure how long it takes things to get pulled down. And so when these subunits are broken down individually, right, you can see that they sediment at a different rate when they're put together. For our ribosomes, we have a 60S subunit and a 40S subunit. Again, the math doesn't add up, right? That makes an 80S subunit. And these are actually really important. We'll talk about this in a lot more detail in the antibiotic chapter, but this gives us is one of the really important targets for certain antibiotics, right? We can target 70S ribosomes, which will typically spare the 80S ribosomes that are present in your cells. We'll talk about kind of a cool exception to this in just a little bit. And it's actually the ribosomal RNA and ribosomal DNA that we use for that phylogenetic identification that I mentioned, right? This is what allowed us to differentiate between the archaea bacteria and the normal bacteria, right? Whatever that means. When I say normal bacteria, that's not necessarily a great term. And we'll talk about more of these identification techniques here a little bit later in the session. Prokaryotic cells also have the unique ability to form endospores. We mentioned those yesterday. And that was one of the problems. Some of those early experiments, they boiled their broth and they still saw growth later. And that's because endospores are incredibly resistant. An endospore, essentially what we look at it is a dormant cell. If you're kind of looking for an analogy, one thing you can compare it to is a seed, right? So you may have, if you like to plant flowers or a garden, you have seeds, and those seeds, you can hold on to those for years, pop them in the dirt, give them some water, they get some sun, and they grow, right? But they've been dormant for that whole time. So that's kind of what an endospore is equivalent to. We see these primarily in Bacillus and Clostridium, right? So primarily in gram-positive cells. There are some, I think there is a genre of gram-negative cells that can also form endospores. Here, though, maybe you've heard of some of these, Bacillus anthracis, right? So this causes anthrax. B. cereus and B. subtilis is what we work with in the lab. Clostridium, I had mentioned Clostridium difficile earlier. For those of you that are in the healthcare field or do end up being in the healthcare field, you'll kind of come across that. If any of you are already CNAs, you may have already seen some of this. And you have to take special precautions when working with these patients. I used to work as a sterile processing technician in college, so we would clean all the instruments and all the equipment after surgeries or after a patient had like something like a pump, like an IV pump in their room, and anything that was from a room that had C. diff, you had to take extra precaution and use a different disinfectant. So in some cases, right, we can consider these kind of almost invincible. All right, let's see. Some of you don't even, Mike, yeah, have any of you seen this movie? Yeah, me either. I hate the Eagles, but I love Marky Mark, right? But the movie's called Invincible, right? But he's actually not invincible. Michael Jackson, some of you probably don't even know who he is, but he still is the king of pop. All right, his last album or one of his later albums was called Invincible. They're not really invincible, right? But endospores, in a lot of ways, essentially are, all right? And again, essentially, we can destroy them, but heat, radiation, antibiotics, dehydration, very, very tough to beat. And so what happens in an endospore, when an endospore forms, is a cell stops growing. Now, what causes a cell to stop growing? Truth be told, one of the big things is they're really great at detecting carbon and nitrogen levels in their environment. If carbon and nitrogen levels are low, right, that means it's not a great environment for them to grow. And so, right, they will start to basically prepare to go into survival mode, right? That's what an endospore is. And so when that happens, you replicate your DNA, right? So your chromosome. So now if you think about it, go back to that E. coli, right? Although E. coli is not forming an endospore, all right? You can kind of think about its size. And now you take a bacteria and you have 2 copies of those circular chromosomes on the inside, all right? And what happens is you have an asymmetric division. And so you start to wall off this chromosome that you've made a copy of. And it starts to become engulfed or encased by the larger component. So you'll start to see the formation of its own kind of cytoplasmic membrane that surrounds it with its own peptidoglycan layer that surrounds it. It then starts to form something called a forespore, right? And this forespore will then get a keratin coat on the outside of it. So keratin is a really tough protein. Sarah, do you remember from AMP1 where we have keratin in our bodies?
We have a lot in our fingernails, right?
Yep. So fingernails, your skin, and your hair, right? It's a really, really tough protein. So one of the things we can do, and we'll go over this in one of the activities that I have for you guys, is something called an endospore stain. And an endospore stain actually requires you to heat up the bacteria to help break down this keratin coat so that the stain can actually get into the endospore, right? So it's really, really tough. So it's that keratin coat on the outside of the endospore that gives it its kind of resistant capabilities, right? So if that forespore is formed, we have keratin on the outside of it. The mother cell then, right, degenerates and the endospore is released. This process takes about 8 hours, which maybe seems like a long time, but on a physiologic level, that's actually a really quick response. And what's even more interesting is that an endospore can become a bacteria in a vegetative state within a matter of minutes. And by minutes, typically 20 plus minutes, but it's really quick. And again, when we say vegetative is we're talking about bacteria, is bacteria that's actively dividing and growing. I know maybe sometimes I keep confusing if you've heard of someone that's in a vegetative state, like in a coma or something, right? This is different. Vegetative in this case means that it's actively dividing and it's alive and doing well. All right. So, what I'm going to do is there's not much here on eukaryotic cells. So I'm going to have you guys kind of look over this maybe on your own. So we'll start with chapter 4 and move into chapter 6 tomorrow. But, a couple big things, that I want to point out here quick, and then I'll give us a little break. Eukaryotic cells can be single cells or multicellular, all right? So when we talk about, we'll talk about algae really briefly. It's a great example of a single cell eukaryotic cell. Protozoa are single cell eukaryotic cells. You are a multicellular eukaryotic cell. We talked about chitin and cellulose in the cell walls of eukaryotic cells that have them, right? Nucleus. I'm crazy here. This idea of endocytosis and exocytosis, remember, because of a cell wall in prokaryotic cells, right? They're not doing this, all right? Again, there's always some exceptions, but we'd say for the most part, we feel confident in saying prokaryotic cells don't take in things through endocytosis. So here you see 3 different types of endocytosis, and then you have exocytosis over here. And you can see digestion taking place inside the cell, whereas in a prokaryotic cell, majority of digestion takes place outside of the cell, right? I mentioned differences in ribosomes, all right? Cytoskeleton, right? This is what helps give your cell structure. Cytoskeleton is also important for, if you go back to our presentation yesterday, eukaryotic cells are a lot bigger, all right? And so you need essentially roads or mechanisms of transporting things from one side of the cell to the other, all right? And the cytoskeleton can provide structure. It also can provide a road or a path for different molecules to travel down. And the cytoskeleton is actually used by eukaryotic cells sometimes too to even allow movement. And there is research coming out now, or people looking at that we didn't think that bacteria had cytoskeletons, and we're now starting to find out that they may actually have some cytoskeleton, at least like structures. My big thing here, and go back and look at this, bacteria do not undergo mitosis. Those of you that had me in ANP1 lab specifically remember that I tried to hammer home the point of mitosis is nuclear division. If you don't have a nucleus, then you don't divide that way, all right? And so bacteria undergo something called binary fission. They don't have a nucleus, so they don't undergo mitosis. We talked about ribosomes. I might kind of mention this tomorrow, but mitochondria and chloroplast actually are very bacteria-like. And so we think that mitochondria and chloroplast may actually be essentially bacteria that a long time ago moved in and formed a symbiotic relationship with eukaryotic cells, all right? And then just some other kind of components of membrane-bound organelles. And this is a really good chart that kind of goes over some of these differences and comparisons, all right? So if you're looking where you should start studying, I always kind of recommend these charts because they kind of lay it out there for you, and then you can go investigate individual components of these charts through your lecture, through other lecture material, and through your textbook. All right, so what we're going to do here is we're going to end. It's 10:06. Let's come back right around 10:20, right? I would say 10:21. That could be too specific for some of you, so I'll say 10:20-ish. So go ahead and shoot for that. Since there's going to be a 15-minute break, I'm going to go ahead and stop this. I will